Okay, we're going to take a look at the types of chemistry that the functional groups in carbohydrate molecules can do. So mostly we'll be looking at the aldehyde and ketone chemistry that takes place, as well as some of the chemistry for the hydroxyl groups. So first of all, we can talk about some of the redox reactions that happen with sugar molecules, and this makes sense, right? If sugars are our primary source of fuel, the way we get fuel is by burning it, just like you could burn any other type of fuel. And so that's an oxidation reduction reaction. So if we take an aldose, we know we can oxidize aldehydes all the way to carboxylic acids. So we're looking, this is some sugar, and this can be the rest of the sugar, whatever it is. What matters is this aldehyde functionality. So we add some sort of oxidizing agent, and we can convert that aldehyde to all the way to the carboxylic acid. So just to give you some terminology, for example, if this were glucose and we oxidized it, it would become gluconic acid. So these are sugar acids when you oxidize that aldehyde. And because of this, sugars, these types of sugars that can do this chemistry are called reducing sugars. So this is a reducing sugar. That's because when it gets oxidized, it causes something else to get reduced. And so this is a good way to differentiate between aldoses and ketoses, because only aldehydes can do this. However, I will say that sometimes those carbonyls in a ketose can move, and you end up with an aldose, and you get the same type of reaction chemistry. But if you wanted to test for an aldose, you would typically do something called a Tollens test. And this works with any type of aldehyde, where you react it with silver ion. That's going to be our oxidizing agent. So that's going to be in the presence of base, like ammonia, and water. So if silver ion is the oxidizing agent, it's going to get reduced. So you end up with your carboxylic acid, H+, and reduced silver ion is silver solid. So that's pretty cool. So what happens as you do this chemistry, you see the silver start to plate out on whatever reaction glassware you're doing the reaction in. We did this reaction when I was in organic chemistry lab, and once we saw what was happening, everybody ran back to their dorm rooms to get whatever glasses they could find and started silver plating everything. It was pretty cool. That was an aside for you. So that's the first reaction we'll talk about. Now let's talk about oxidation at the primary alcohol. So that's the alcohol, typically at the other end of the molecule. So if we take glucose with its aldehyde on one end, then I'll just skip all of these chiral carbons in the middle. At C6, here's our primary alcohol. So this guy right there, that's a primary alcohol group. So now we can also oxidize this one to the carboxylic acid. And you may start wondering, well, how is there any sort of control in these reactions? And remember, when we're talking about chemistry in the body, we're using enzymes to facilitate those reactions. So they're very specific in terms of how they bind their substrate and where the chemistry takes place. So even though we can get oxidation of a sugar molecule at both ends, the enzymes are very specific in controlling which end that chemistry happens at. So the next type of chemistry we want to take a look at is the formation of substituted derivatives. So even though we said in the beginning, substituted derivatives, that we're talking, when we talk about sugars, we're talking about molecules with carbons, hydrogens, and hydroxyl groups. We can also add other things to it. So for example, we can deoxygenate sugars, like in DNA. We can add amino groups to sugars, like in the bacterial cell wall. So let's take a look at that. So this is figure 7-9 on page 249 of your textbook. So for example, you can see glucosamine, where we've added amino group in place of one of the hydroxyl groups on the sugar ring. What else is interesting here? Here's muramic acid with, again, an amino group and now an R functionality on that ring. You can phosphorylate sugars. Glucose 6-phosphate is a common intermediate in metabolism. So you can get all of these different derivatives of these molecules. And as you read through different papers that involve studying carbohydrates or carbohydrate-containing molecules, you'll see all sorts of interesting nomenclature, like this carbohydrate has a GLICNAC, so really cool acronyms and things like that. So when you start hearing words like this, those are going to be sugars that are substituted in some way with a different functional group. One of the biggest types of chemistry, though, that we're going to be concerned about is the polymerization reaction of sugar molecules, which means they can form a glycosidic bond. So that is a bond between two monosaccharides. So it is the, I can't remember the word that starts with an A, but it means it's the cousin of the amide bond, which links two amino acids in a peptide. So it will link two amino acids. Oh, analog. I actually have that written in my notes. It's the analog of the peptide bond. So let's take a look at what that chemistry looks like. So one of your figures in your textbook. So this is figure 710 from your textbook. So this is the reaction of glucose, alpha D-glucose, and beta D-glucose. So we're talking about the reaction of two anomers of the glucose molecule. So they differ in their stereochemistry at the anomeric carbon, which, remember, is this one right here I'm showing in green. So hopefully you can tell that this one is alpha because the OH group and the C6 are on opposite sides of the ring, whereas this one is beta, where they are on the same sides of the ring. So as we do the chemistry to form this glycosidic bond, we're going to have a nucleophilic attack here by this oxygen atom all the way over at the anomeric carbon. And during that process, we are going to do a condensation reaction, which means we're joining two molecules together, we're condensing them, and we're going to lose a water molecule with the atoms shown here in pink. So right here is our glycosidic bond. And it's going to matter, and we're going to talk about how you describe this bond, because it's going to matter if you form that sugar bond between two anomeric carbons or an anomeric carbon and some other hydroxyl group, which you can see on a sugar molecule. We have a lot of options for that. And another interesting thing that happens is that if you block the anomeric carbon, as we have done here, right? So this anomeric carbon is no longer part of a hemiacetal. It is now an acetal. It's connected to two ether groups instead of an ether group and a hydroxyl group. When you block this anomeric carbon, you no longer have a reducing sugar. So if the anomeric carbon is in the glycosidic bond, then it does not reduce molecules anymore. So you can change the chemical properties of your sugar by hiding the reducing ends. Now, in this case, at the other end of the molecule, here we still have a free anomeric carbon, right? So the bond here was formed between an anomeric carbon and just a regular old carbon. And so this anomeric carbon over here is still free, so it's still a reducing sugar overall. So we have one reducing end in this sugar. This is going to be important physiologically because the enzymes that break down these sugar polymers, particularly starch, recognize differences between reducing sugars and non-reducing sugars. That's how they're able to break down glucose break down starch and release glycogen and release free glucose, which is very important in your fight or flight response. You know, if you're being chased by a bear in the woods or something and you got to book it out of there. So let's take a little bit of a closer look at some of these disaccharides and talk about how we specifically name the glycosidic bond that's linking the two molecules together, because the nomenclature has to tell us the name of the sugar, the stereochemistry of any anomeric carbons participating in that glycosidic bond, and which carbons in fact are participating in that glycosidic bond. So let's take a look here. Okay, so here's 3 examples of disaccharides: lactose, sucrose, and trehalose. So let's take lactose as our first example. So when we describe this glycosidic bond, and you're not going to have to do this part where you've got to name all those complicated sugars. Oops. So you don't have to worry about this. That gets really complicated. And in fact, you're not even going to have to worry about this either, where you have to know the abbreviations and identify the specific type of sugar, like what its actual name is. But what I am going to hold you accountable for is figuring out this part right here. So being able to look at the molecule and describe that linkage that's taking place. So what we see here, first of all, is the 1-4. That's identifying which carbons are participating in that glycosidic bond. So it's a glycosidic bond between carbons 1 and 4. And then if there's stereochemistry, if we've got an anomeric carbon, that's going to matter. So we have to identify, for example, in this case, that this was a beta sugar, right? So this was the beta anomer, and you can tell that because this fact that this bond is going up means the hydroxyl group was originally pointing up on the ring, which means it was on the same side as this C6, which is a beta anomer. So that became beta 1,4 glycosidic bond. For carbon number 4, it's not necessary to give the stereochemistry there. First of all, that's not a carbon that participates as an alpha and beta anomer. That's only determined by the anomeric carbon, which in the second sugar is still over here, carbon number 1. So the stereochemistry at that position is included in the actual name of the molecule, which was glucopyranose. But I'm not going to hold you responsible for saying, oh, that's glucopyranose. Okay, I'm just going to hold you responsible for identifying the linkage of the bond. What you also should identify here is that since carbon number 1 of this second sugar is still got its hemiacetal, this is still a reducing sugar. Okay, so let's take a look at the second one. In sucrose, we've got a bond between carbons 1 and carbon 2, and both of these are anomeric carbons now, right? So both of these are where the hemiacetal or the hemiketal, in the case of the second sugar here, where that actually formed at. So if we look at this, we can see that in the first one, this bond was pointing down. So that means the original hydroxyl group on that anomeric carbon was on the opposite side of the ring of C6. So that's an alpha anomer. So this is an alpha 1. And this was a beta anomer on the other hand. So now you've got to really keep the structure of the sugar in case. So here the bond is pointing down. So the hydroxyl group that was on that anomeric carbon was pointing down. And in this case, the C6 is also pointing down. So those two groups are on the same side. That makes this a beta anomer. So it's a beta, and that's carbon number 2 because that's where the carbonyl was. That was the anomeric carbon. And so this is an alpha 1 beta 2 linkage. Since both of the carbons participating in these bonds were anomeric carbons and they're both blocked now, sucrose is a non-reducing sugar. Then let's take trehalose, our last example here. So we've got a bond pointing down, so a hydroxyl group pointing down, a C6 pointing up. That's an alpha 1 carbon participating in this glycosidic bond. And then if we look over here, we have the bond pointing up. Ooh, here's C6. You got to find it. C6 is pointing down. So those are on opposite sides of the ring. That's an alpha stereochemistry as well. That's carbon number 1 in the ring. So this is an alpha 1 glycosidic bond. Both anomeric carbons are participating. So then this also is now a non-reducing sugar. So we change the chemistry by blocking those anomeric carbons. So I'll expect that you can figure out which carbons are which in this ring so that you can number them properly. And you've just got to look for, just to point this out again, that the oxygen in the ring was a hydroxyl group that reacted with the carbonyl carbon, so you can identify the anomeric carbon immediately, and then you can backtrack in terms of counting to figure out what the carbon numbers are.